Okay. Well, let's go and get rolling for the day. So, announcements. Quiz 7 is going to be due on Friday, and your problem set's going to also be due on Friday as well. So, keep those things in mind. The problem set right now, hopefully I get through this one slide today. If I can get to this one slide and through it today, we'll be fine with the problem set. Okay. I don't think we're going to finish our lecture today, but I'll let you know if we're going to have to delay the problem set. I hope to not do that, because then that would mean it would be due on the same day your exam is due, and I don't want to do that. Okay? I'm pretty optimistic that we'll get through it. Okay? So our riddle here, the man who built it doesn't want it. The man who bought it doesn't need it. The man who needs it doesn't know it. What is it? Coffin. A coffin. Right? Exactly right. Morbid, but true at the same time. Okay, now let's move here. Not that one. Not coffee. I know. That coffee does not work for that. Right? Coffin. Yeah, coffee is definitely not the answer on that one. There are some people who don't want it, but okay. So obviously Monday we did our group work assignment. And I've got those grades. All you need to do is just post them and also put those group work assignments in MRB 111. I hope to do that this afternoon sometime, okay, or tomorrow. So hopefully this afternoon. And it looked like people did pretty well. So I was happy about that. That one is not necessarily the easiest one of all of them, for sure. And I was happy to see that results looked pretty good. So on Friday, this seems like eons ago, we left off essentially right here. Hopefully you have a little bit more context for that. I talked about this protein called glycogenin. All right, and glycogenin forms the core of these glycogen granules. And glycogen is this protein that is a dimer that will essentially prime the synthesis of glycogen, of the glycogen granule. And it has this, coming over to this part of the slide, we didn't really talk a little bit too much about this, but it has this tyrosine residue on it that's actually shown right over here and also right here, because it's a dimer. And that tyrosine has a hydroxyl group on it that will attack the first carbon of this molecule UDP glucose. You have now hopefully context for UDP glucose. UDP glucose is made during the glycogen synthesis process. We'll get to that enzyme here in a little bit. But nonetheless, this enzyme or this protein has this hydroxyl group on its tyrosine 194 that attacks that first carbon. And that then makes essentially this, splits off UDP and makes the glycogenin with a single glucose on it. Okay. After that, this is the fourth carbon of that glucose. That glucose will attack then the next first carbon of the incoming UDP glucose, and it will do this 8 times. Right, and each protein, because this is a dimer, will do that 8 times, and that's what I essentially said, that they would lead to this 8-mer of glucose residues. Okay. After that point in time, glycogen synthase will take over and start adding to that. And that's where I had you pick up in terms of the group work assignment, okay, is essentially right here. Okay. And then you're to add those 11 glucose molecules. Now, let me go ahead, we'll come right back to this slide here in a little bit. That's the breakdown. But let me go ahead and go right to this slide and just review hopefully what you learned on Monday in the group work. So we were just talking about this molecule glycogenin. Okay, that's what we were just looking at on that last slide. Okay, and there's that tyrosine residue that's going to make that 8-mer. Right? Now, glucose synthesis really begins at glucose 6-phosphate. So glucose enters into the cell, whether this be skeletal muscle cell, which can make glycogen, or this be the liver. Either way, both of those will trap that glucose using hexokinase. That's the trapping reaction. Glucose 6-phosphate then is held in equilibrium with this enzyme, phosphoglucomutase. And phosphoglucomutase will simply move the phosphate from the 6th carbon to the 1st carbon, okay, making glucose 1-phosphate. That becomes the substrate for this enzyme that will make UDP glucose. UDP glucose is made by the enzyme UDP glucose pyrophosphorylase. Okay, fun to say, fun to spell. UDP glucose pyrophosphorylase. And it's going to require not ATP, but UTP to do this, which is an ATP equivalent. Again, from an energetic standpoint, UTP and ATP are essentially the same molecules. Obviously, they're different from a genetic standpoint, but energetically speaking, they're essentially the same molecule. And that UDP glucose pyrophosphorylase will use that UTP. It'll essentially make UDP glucose here and split off pyrophosphate. Let's try that again. Pyrophosphate. Okay, there. I can make it through the rest of it. Okay, so pyrophosphate. And that's going to be hydrolyzed by this enzyme pyrophosphatase. Now, to 2-PI. That entire process and the use of UTP makes this enzyme here have a delta G that is much less than 0. So it's irreversible. Okay. Now here's what's tricky about this. That is not the key regulatory step of glycogen synthesis, despite the fact that it's irreversible. Okay, it's not the regulatory step, and the reason is because UDP glucose, that is a substrate for lots of different enzymatic activities in the cell. Okay, so the UDP glucose pyrophosphorylase, while we're showing it as a part of the glycogen synthesis pathway, that enzyme is found in other pathways as well. So it's not a regulatory enzyme, certainly for glycogen synthesis, okay, despite the fact that it is irreversible, okay, which would suggest, based off of what you've learned, that it would be that. Okay. Now, this next step that they're showing here, right, this is essentially the priming. So priming. And glycogenin does that. Okay. And once it gets that 8-mer sticking out, that's when we get to this enzyme glycogen synthase. Now glycogen synthase is, delta G is less than 0, and this is the key regulatory step of glycogen synthesis. And we're going to talk about, of course, regulation of glycogen metabolism, and glycogen synthase is going to play an important role in that. Okay. And this is just going to take those UDP glucoses and just going to add them to that primer event. It's just going to keep adding. And every time it adds one, it splits off UDP. Okay, and just makes this molecule. Right. The last thing that we see here, okay, is this, and that is that usually, how many do I get? 1, 2, 3, 4, 5, 6, 7. There is this branching enzyme. Okay, they're not really showing it here. They only show a movement of 5. But the branching enzyme will usually grab 6 to 7 glucose molecules and move them upstream. Okay, and it will make an alpha-1,6 bond when it does that. Okay. Glycogen synthase makes alpha-1,4. Glycogenin makes alpha-1,4. Okay. The only one that makes alpha-1,6 is the branching enzyme, or 4,6 transferase. Okay, it would be another name that it goes by. And then, of course, the process is just going to keep going, okay, until you get this, what would be considered fully developed glycogen granule. And that fully developed glycogen granule will be highly branched, right, and it will have over 100,000 different glucose molecules in it. Now, to give you an idea of the importance of storing glucose in a granule, if that granule was hydrolyzed instantaneously in the cell, it would lead to molar concentrations of glucose in the cell. And then water would rush into the cell and the cell would go pop. Okay, so it's important that the breakdown of the glycogen granule is regulated and that the glucose that's coming off of it is used. It's also important that this synthesis process is regulated and that, of course, we're storing glucose in this way so we don't get these really high concentrations of glucose in the cell. Okay. The other aspect of this is that then in the branching aspect of it, it leaves these lots of these ends that are called non-reducing ends that then the breakdown apparatus can easily access all of these things, so it can break it down fairly quickly if needed. Okay. Yes, question?
So since each of the chains
Yeah.
long, does like glycogen synthase synthesize longer than 14, and then the branching enzyme like takes the excess?
Yeah, okay, so the question is an interesting question that I don't know if I know the exact answer to, okay? But the question is, does glycogen synthase make something that's longer than, like, let's say 14, and then the branching enzyme just moves them as it goes? My intuition would say yes. I don't think there's anything in glycogen synthase that would know what is downstream of it, okay? I don't think it's that big of a molecule. It could be. But I think it's the branching enzyme that really determines the length of those branch points, 12 to 14 residues. And it just keeps acting. Every time it gets a little bit too long, it just moves 7 to somewhere else. And then glycogen synthase keeps going. So, yeah. And the other, I had a discussion with one of the groups, at some point in time, the glycogen granule stops growing. It becomes big enough and it just says, this is no more, right? And what are the signals that tell these things to stop doing that? I don't honestly know. It might just simply be, some of the regulatory steps that we're going to talk about. But yeah. Question?
I may have missed this if you said it here. The branching enzyme, it says the 4-6 transferase. Is that just the alpha 1 to 4 to alpha 1 to 6?
Yep. Yeah. So another name for this branching enzyme is 4-6 transferase because it's taking alpha 1 to 4 to alpha 1 to 6. Yeah. Okay. Other questions? Yeah.
It's like when these enzymes have to be fully localized in the cell, so like that's probably how they can be regulated. Is like.
Yep. And we're going to look. Yeah, so when we get to regulation, we'll actually look at how they're localized. So if you didn't hear the question was, are these enzymes localized in the cell? And they are. So we'll talk about scaffolding proteins that are part of the glycogen granule that bring all this stuff together so that it can build this granule all in one isolated portion of the cell. Yep. Okay. Okay, now let me just step back and look at glycogen breakdown. That's synthesis. Let's review the process of glycogen breakdown. And of course, in glycogen breakdown, you're going to start with the glycogen granule and just essentially get Pac-Man molecule, okay, enzyme that just comes along and starts to chomp these things away. That Pac-Man, okay, is glycogen phosphorylase. And it will just start at non-reducing ends, and it will just start removing these as glucose 1-phosphate, okay, from that non-reducing end. Now, what they're not really showing here, and I required you show this on your group work assignment, is they're not showing that inorganic phosphate has to be used in this process. So they break off 10, I think. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Yeah, so the stoichiometry should be 10 of these would have to go in. And every time glycogen phosphorylase removes a glucose molecule from the granule, it requires an inorganic phosphate. Okay. Now, at some point in time, Pac-Man is going to run into something that it can't do anymore. Pac-Man is glycogen phosphorylase, if you're following my video game analogies. Okay. I grew up in the 80s, so Pac-Man was it. I mean, my kids play these games like Fortnite and stuff, and it just makes me, I get motion sickness watching it, honestly. I can't even, and they make fun of me and stuff like that, and it's okay. And so then, but then I might play them in something real like ping pong or pool or something like that, and I get to make fun of them after that. So that's fun too, okay? Either way, someone gets made fun of, okay? Anyway, so Pac-Man, chomp, 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 at some point in time is going to run into this alpha 1-6 linkage, okay? And honestly, glycogen phosphorylase, as it moves down the glycogen granule in these chains, when it gets about 4 glucoses away from that, 3 or 4 glucoses away from that alpha 1-6, it just grinds to a halt. It physically is obstructed by the alpha 1-6 covalent bond. And so it will stop, it will just fall off, and that's where this enzyme, not the branching enzyme, but the debranching enzyme has to work. All right, and what we see here is this transferase activity of it. Now, the debranching enzyme has 2 activities. There's the transferase activity, and it will take these guys, and it's going to move them to here, and that's what you see here, and that's the transferase activity. Now, it leaves this alpha 1-6 bond here, so we still got alpha 1-6 here. And the same enzyme, the debranching enzyme, has a glucosidase activity that will just simply remove that as glucose. Doesn't need phosphate or inorganic phosphate to do this. It just breaks the bond and releases glucose. Okay? And then the cell will do something with glucose, right? I mean, more than likely in the liver, it might export it or you know, it could convert it to glucose 6-phosphate, either one. Okay. Right. All right, and then, of course, this is going to go on, and you'd have then, once you get to this point here, now glycogen phosphorylase, okay, is going to occur again, and it will just start chomp, 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 chomp until it runs into another branch point. Yep.
So if glycogen phosphorylase is simply reversing the buildup, how do you regulate that so you're not just constantly building up and breaking down glycogen?
Yeah, well, okay, so the question is a really good one with a very complex answer that will take us a couple days to get through. Okay, so the question is, if glycogen phosphorylase is simply breaking it down and glycogen synthase is simply building it up, how do we know we're not just spinning our wheels, right, essentially? And so we'll look at that. There's a reciprocal regulation of those two. Okay, it's similar to glycolysis and gluconeogenesis, where we shut down one and we upregulate one. Okay. Now, having that question, let me take the opportunity to say that this is the key regulation step here, glycogen phosphorylase, Pac-Man. Okay. And in our discussions for the next, well, the last portion of class today, we'll look at that. Okay. Now, one last thing I guess I should say. Okay, these glucose 1-phosphate molecules, these are going to be converted to glucose 6-phosphate by the enzyme phosphoglucomutase. Same enzyme that in synthesis converted glucose 6-phosphate to glucose 1-phosphate. That's a reversible enzyme. It can go in either direction. Okay, it's just responding according to Le Chatelier's principle. Okay. Questions? A review of essentially, hopefully, what you learned on Monday. Okay? Now, regulation. Fun stuff. Let me start with a question. We got to go big picture here. Okay? Just like we did for glycolysis and gluconeogenesis, good idea to take a step back, think about big picture first, and then go forward. Okay, so I'm going to give you some time to think about this. We've got all these different signals: insulin signaling, glucagon signaling, epinephrine signaling, and then finally high AMP. Okay, and AMP is adenosine monophosphate. Okay, so the breakdown product of ATP. So you've got these 4 different signals, all of which can affect glycogen synthesis and breakdown. Okay, so what I'm asking you to do is predict these effects, okay, as a whole. And so I'm going to give you a minute 45 to look at those different responses. Is insulin signaling going to lead to glycogen breakdown? Or is it going to lead to glycogen synthesis? Is glucagon signaling going to lead to synthesis or breakdown? Is epinephrine going to lead to synthesis or breakdown? Is high AMP going to lead to synthesis or breakdown? What do you think? Right? Find the right response in that forest of responses, and let me know what you think. Okay? And your marks, get set, go. Minute 45. Save changes. Why not? Okay. Okay, about 15 seconds left. So submit an answer. Okay, let me check this out. Show results. Okay. A smattering of answers here, but 48% of you landed on the fact that insulin signaling should lead to glycogen synthesis. And the other three, glucagon signaling, epinephrine signaling, and high AMP, will lead to the breakdown of that glycogen granule. Okay, so let me go ahead and move here and give you the answers for this. Okay, so there are really three different states that we would consider here. A fasted state of the organism, okay, a stressed state of the organism, and by organism I mean like a mammalian organism like you and I, and then finally a fed state. Okay, let me go ahead and start all the way on the right. Okay, fed state leads to insulin secretion, especially if the fed state had a carbohydrate-rich meal. All right, so we'd have insulin spike in the bloodstream. And as we're going to find out, this is going to lead to glycogen synthesis. Okay, again, big picture here. If there's a spike in blood sugar because you just ate a meal, then especially the liver should be producing glycogen. It should be taking that in, not using it for energy needs, but rather producing glycogen. Okay, and so this is really important in the liver. Okay. To a certain extent also, you'll see it with the muscle cell, but certainly the liver will see that. Okay. Now, moving to the left here, all the way to the left, let's go to this end here, a fasted state. Fasted state is going to lead to an increase in glucagon. Okay. Blood sugar is dropping now, or has dropped. Pancreas detects that. Liver will detect that because glucagon signaling will occur, and this is going to lead to the glycogen breakdown. Right. Very important that the liver be able to excrete glucose. Okay. And so that's going to be very important for the liver. This is really the liver only. And so I should put over here and muscle. Okay. Well, this is liver only because glucagon really doesn't affect muscle metabolism. There's no receptor for glucagon. Okay. Well, that would be glycogen breakdown. Of course, this is also going to, as we discussed, lead to gluconeogenesis, right? So the liver is just getting rid of glucose at this point in time. Okay. Stress state, this would be epinephrine. Or just simply high levels of AMP. And this is going to be really important in muscle cell. This will lead to glycogen breakdown. So 48% of you were indeed correct. Okay. This will be very important in muscle. Okay. Epinephrine signaling, of course, it prepares us for the fight or flight response. Mountain lions. That's all I'm going to say. All right. Or high levels of AMP mean that you're just exercising, something like that, right? In particular, in the muscle cell, that's going to lead to the need for glucose. Okay. And it's very likely that the muscle cell actually can't be absorbing glucose at this point in time. Okay. If insulin signaling is not present, it might not have the GLUT4 transporters in the membrane, so it can't really absorb its own glucose from the bloodstream, or very little of it. So it has to have a supply of that ready and available, and that would be in glycogen. Okay, 2% of the mass of the muscle cell is glycogen. 10% of the mass of the liver cell is glycogen. Okay, you have more muscle than you do liver, though, so the majority of glycogen in the body is found in the muscle. Okay, right. Right, so that hopefully gives you the big picture. Now we need to get down into the nitty-gritty details of how these signals, how AMP, and all these kind of things lead to this. All right, so here's how we're going to approach this. All right, so again, 48% of you were correct in this, and here's how we're going to approach the nitty-gritty details of this. We're certainly going to have to look at regulation of glycogen breakdown and synthesis in the liver and the muscle separately. So those will be our last couple of slides as we look at that, because they are different, right? But there are going to be what I am labeling here principal parts of the regulation of these pathways that would be found in both cell types. Okay, the first one would be essentially this, and that is phosphorylase, or what we have called glycogen phosphorylase. Right, this is the molecule that carries out this, right, where we go glycogen in to glycogen in -1. And glucose 1-phosphate comes out. Of course, inorganic phosphate goes in. That's what this enzyme will catalyze, glycogen phosphorylase. It is the key regulatory step of the breakdown pathway. Okay? Now, it exists in what's called phosphorylase A and B. Okay? That's how the field has denoted these two forms, A and B. The B form is less active. The A form is more active. A for active, I guess. B for because. Okay, I don't know. B for it comes after A. I'm not sure what. Okay. Now, one of the ways it can go from B to A, going in this direction, is to be phosphorylated. There are these two serine side chains. Okay. It's actually a dimer, so they're the same serine side chains. But they can be phosphorylated, and that will make this active, right? So again, sunbeams would be our denotation of active. Okay, phosphorylation. Now, it can also then, of course, be inhibited going in this direction simply by removing those. Okay. What are the enzymes that do this? Well, this enzyme here, it's called phosphorylase B kinase. Another term that it goes by is glycogen phosphorylase kinase. Which actually, I think, makes sense. Glycogen phosphorylase kinase. Adds a phosphate group to the phosphorylase. This one, I think the nomenclature on this that the book uses is not right. PP1 is right, but phosphorylase A phosphatase is not really quite accurate. In our upcoming slides, I'm going to call this PP1C. Okay, and I'll be honest with you, regulation of PP1C and PP1C is not easy to understand. Okay, I'll try and keep it out of the weeds on that, but it can be fairly complicated. Okay, but PP1C is going to remove that. Okay. So that would be one aspect of the regulation of glycogen phosphorylase. Phosphorylated, it becomes active. Dephosphorylated, it becomes less active. Now, understand as well that there are allosteric regulations of it as well, of phosphorylase, glycogen phosphorylase. Okay, and in the liver, it is inhibited by glucose and glucose 6-phosphate. That should make sense. It's a feedback inhibition. Right? If glucose levels are high in the liver cell, it should feed back and inhibit something that's breaking down a glycogen granule. Okay, and that's what's being stated here. Okay, skeletal muscle or muscle. Okay, calcium is a really important regulator of this. Okay, and calcium is going to positively regulate this. Okay. As well as ATP and AMP. Okay. And actually, I got ahead of myself here. Calcium, I'm going to give myself a red check minus here. Calcium is my next slide. Sorry. Okay. Cross that off. Calcium does not allosterically regulate glycogen phosphorylase. It regulates glycogen phosphorylase kinase. Okay. So, but ATP and AMP do. Okay. Sorry, red check minus. I know. It's a Wednesday. We're making it through. It's a overcast Wednesday. Okay. Questions on that? Yeah.
Would one be positive and one be negative regulation?
Oh, yeah. Good question. Okay. Yes, they would be. Negative regulation. I got so discombobulated by my mistake that I forgot to give you positive and negative. So ATP will negatively regulate, allosterically negatively regulate this, and AMP will positively. Okay. Again, that should make sense. AMP needs to be a signal to break down, right? ATP is a signal that, no, we don't need to break this down. Okay. Other questions? That's glycogen phosphorylase. Now, our next slide, we need to look at this and this. So let's go to that. Oh no. I'm so anticipating that next slide that I forgot this slide. Glycogen synthase. Let's do glycogen synthase, and then we'll come back and do that, okay? Okay, so glycogen synthase, the other key component, right? So glycogen phosphorylase, the antithesis of that, or the counterpart to that, would be glycogen synthase. Glycogen synthase carries out, of course, the addition of glucose to the glycogen granule. Okay, it uses UDP glucose as its substrate. Now, this is, again, we have our sunbeams. Active, the glycogen synthase A, active, dephosphorylated. Okay, so when it's dephosphorylated, it's active. Now, here's what's really fun. Okay, this can be phosphorylated. And there are 9 serines that can be phosphorylated. Nine total. There are 11 kinases that do this. 11 that we know of. That's not fun. Okay. We're not going to look at all 11 of them for sure. Okay. We're going to look at a couple of them. One of the ones that we will certainly look at will be glycogen synthase kinase. Okay. GSK3, glycogen synthase kinase, which is actually listed here. Okay. AMP kinase, which we've talked about. Okay, adenosine monophosphate, or AMP activated kinase will be one of them. And then finally, PKA will be another one of these. Okay. PKA is, of course, activated by epinephrine or glucagon signaling. Okay. All of those will lead to the phosphorylation of this. They're only showing 3 phosphate sites, but those it becomes more inactive the more phosphorylation it has undergone. Okay, to the extreme where there would be 9 total phosphates on it, and it would be at a snail's pace in terms of enzymatic activity. Okay. And then moving in this direction, dephosphorylation is again accomplished by PP1C. Okay. Your book mentions CK2 and the phosphorylation of this kinesin kinase. We're not going to mention it. Okay. But PP1C, the same thing that we talked about over here, dephosphorylates here, dephosphorylates this. Okay. Activating one, inhibiting the other. Okay. Questions on glycogen synthase? Awesome. Let's do this. Let's move on. Now here's that slide that I so couldn't wait to get to, and I'm not sure why, but I couldn't wait. Okay, I kept on thinking this is my next slide. So here it is. So this just gives you again more of these parts. Okay, phosphorylase kinase. Another name by this again would be glycogen phosphorylase kinase. Okay, This enzyme is actually fairly complicated. This is a figure that's not in your book. It's a tetramer of tetramers. Okay, and so let's highlight one of these. Okay, it has an alpha, and for some reason they don't have the alpha labeled. But it's alpha, beta, gamma, and delta subunits. Okay? And that's what you're seeing there. And then there will be 4 of those. So in reality, the enzyme has this kind of structure. Alpha 4, beta 4, gamma 4, delta 4. Okay? Now alpha and beta, okay, these can be phosphorylated. Okay? And when they're phosphorylated, put a P with a circle, they're active. When they're dephosphorylated, okay, it inhibits glycogen phosphorylase kinase. So alpha and beta activated by phosphorylation, inhibited by dephosphorylation. And you'll see that in upcoming slides. The delta, okay, this guy, this is actually a calmodulin protein, which can be activated by calcium. Okay. And then finally, gamma is the catalytic subunit. Okay. Now, glycogen phosphorylase kinase, okay, can be phosphorylated, and when it's phosphorylated, it can phosphorylate glycogen phosphorylase, and that will activate glycogen phosphorylase. You'll see. I've got a whole slide for it. It's going to be lots of fun. Okay? Or it can be activated by calcium. So that's an allosteric activation of it. We'll get to that later on. Catalytic unit will be that gamma subunit, and it's going to be what's going to phosphorylate things. Okay? Now, that's glycogen phosphorylase kinase. This over here will be best called PP1C. Okay? And PP1C, the regulation of PP1C is not easy to describe. Okay? And the reason is because PP1C is one of several isoforms that can be found in a cell. And those isoforms can interact with what would be called regulatory subunits or even targeting subunits. Okay? And the number of regulatory / targeting subunits is in excess of 20. Okay? I looked at it again this morning and I found at least 20. And that was just with the limited amount of time that I had to look into it. Okay? I'm sure it's a growing number. PP1C is a pretty complicated protein. It can be targeted to different parts of the cell. It is what would be considered a serine-threonine phosphatase. So it will remove phosphates that are found on serine or threonine side chains. Okay? So we'll talk about how it's regulated with regard to glycogen as we move forward, but let me just go ahead and give you the organization that it's found in. So this blue thing here, this is a figure that's not found in your book. This would be, I mean, it says glycogen, right? It's just the granule. And obviously that's a very, very poor rendition of what the granule looks like. Blue sphere, though, stands for granule. Now, in that, interestingly, there's going to be these targeting proteins, glycogen targeting proteins. So let me highlight this. GL is a glycogen targeting protein. And it's a scaffolding protein. And GL, L subscript L, stands for liver. Okay? It's scaffolding, meaning that it's not a protein that has enzymatic activity, it just has a structural role. When it's there, it has binding sites for lots of other enzymes, and it brings all those together into one central point so that regulation and catalytic efficiency can be all put into just one part of the cell. In this case, glycogen. Okay? So glycogen targeting protein or glycogen targeting subunit, that would be another way to term this. Now, that, okay, has binding sites. Okay, so it binds glycogen synthase here, GS. It will bind to glycogen phosphorylase. Shown here, this guy. Okay. It will bind to glycogen phosphorylase kinase, shown here, which is also this guy over here. And then finally, to PP1C, which is really easily labeled. Okay. And so it organizes all those things. Now that doesn't tell you how PP1C is regulated. We'll get there. Again, it can be fairly complicated. Okay. But we'll leave it at that. It's there. So those are our parts: glycogen phosphorylase, glycogen synthase kinase, and glycogen phosphorylase kinase. And throw in AMP, K, and pKa for good measure. And you think to yourself, that's a lot of abbreviations. Exactly. Okay? That's biochemistry. All right. All right, now let's take a step forward. This is the slide that I wanted to get through. If we can get through this slide, you can do the problem set. Okay? And I've got time to do it. So if you can get through this, we're going to be great. I'm going to get through this. We're going to be great. This is going to be fun. Now, where do we start with this whole thing? And you think to yourself, boy, that's just a lot of boxes. And you have red and green boxes, and I will be drawing plus and minuses in these, but red means inhibited, green means activated. Okay? Again, I'll add plus and minuses for those that you have difficulty differentiating between red and green. But let's do this. Let's start here, glucagon. Glucagon activates pKa, as we know. Several steps. We don't need to talk about it. Okay? Now, here's what pKa is going to do. Okay? First of all, pKa, as you know, and understand that this is in the liver. Okay? pKa, as we know, is going to activate gluconeogenesis. So we've talked about that. Fructose 2,6-bisphosphate is important in that whole process. Done. pKa, remember, is a kinase. And so it's going to phosphorylate things. Okay? It's going to do stuff. And so this, first of all, is going to do this. It's going to phosphorylate glycogen phosphorylase kinase, or phosphorylase kinase. It's going to put two phosphates on it, right? On the alpha and the beta subunit. You need 2 ATP to do this, because that's where kinases get their phosphates from. Okay? And that's going to activate phosphorylase kinase. Now, phosphorylase kinase is then going to phosphorylate. Again, it will need 2 ATPs to do this. Glycogen phosphorylase. Okay? Put a phosphate here. And on the other one, remember those are the serine residues that get that. That will activate that. And then that will go, it's the Pac-Man molecule. Remember, it's going to take glycogen N+1. And it's going to make glycogen from it. And out comes, let's go down here. Out comes glycogen 1-phosphate, which will then be converted to not glycogen 1-phosphate, glucose 1-phosphate, sorry, which will be converted to glucose 6-phosphate, which will then go on and be converted to glucose more than likely, because this is at the liver. Glucagon signaling says to the liver, send glucose. Okay? So glucose. Okay. Now, if we're not going to stop there. But if we stop there, you'd have enough to do problem set 2. Okay. All right. Just in case you were scared. Okay. But we'll keep going, obviously. We need to keep going. Now, pKa, this is going to also be fun. pKa goes over to this side. And pKa is one of the enzymes that will phosphorylate glycogen synthase. Remember, glycogen synthase has like 9 different phosphate sites that it can have on it. So pKa will start to phosphorylate that, and it's going to inhibit that. Okay. Glycogen synthase kinase will also, which is active here, okay, will also do this. Okay, so you'd have to have X-ATP going to X-ADP. Okay. And of course that means glycogen synthase is not going to be making glycogen. Going back to the question that was asked earlier, how do we make sure that we're not building glycogen and breaking down glycogen at the same time? Exhibit A. Okay. Glucagon signaling leads to the activation of pKa, which phosphorylates phosphorylase kinase, which phosphorylates glycogen phosphorylase, which starts to break down glycogen. pKa also phosphorylates glycogen synthase, which is also being phosphorylated by glycogen synthase kinase, all leading to the inhibition of glycogen synthase. Don't you love biochemistry? I mean, you know, how many other classes do you hear a sentence like that? I think I said phosphorylate like 7 times in that sentence. Okay, and I mean, the amount of words that I use, like different words, there's only like 10 different words I use. I just put them in different order, right? Okay. 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 Now, that's going to lead to glycogen breakdown. Okay. What about glycogen synthesis? Let's do this. Because, well, let's first of all do this. Let's look at insulin. Insulin signaling will do this. It leads to the activity of AKT or PKB, remember? Okay, that phosphorylase, not that phosphorylase, that kinase. This will phosphorylate glycogen synthase kinase. Okay, so insulin is the opposite signal, says don't send sugar, absorb sugar, start to make glycogen, right? So that's going to phosphorylate glycogen synthase kinase, inhibiting it. So that will be inhibited. Oh boy, I need something that shows inhibition. There we go. Negative. There we go. Okay? And then if this is active, let's come down here. If this is active, that's not very good. Let's put a plus here. If this is active, PP1C is going to do this. It's going to do this. And it's going to do this. It's just going to break off phosphates. Okay? X phosphates. This would be, coming back here, would be 2 phosphates. And here it would be 2 phosphates. Okay? And it's just going to, PP1C is just going to reverse everything that pKa just did. Okay? Now you're all sitting there saying, yes, I see that, because your green boxes tell me so. But why is PP1C active again? That doesn't make a lot of sense yet. And we don't have the time today to discuss why PP1C would be active. Okay? So we're not going to do it. So we didn't quite finish this slide, but we finished it to the point where you can do problem set 2, hand it to me at the beginning of Friday's class, and then we can move on and finish our discussion on glycogen metabolism, be ready for the exam on Monday. And you say, oh, goody. Okay? All right. I will see you on Friday. Say it again, what's that?
So does the pKa also activate GSK3, or are we
No,
just assuming like that's
we haven't talked about it. I'll mention that. Yeah, it's a good question. I'll mention that on Friday. So GSK regulation is really complicated. It actually has over 100 different targets in the cell that it will phosphorylate. There's an estimated that it's 500 that it will target. So the regulation of it is crazy. So we aren't going to really talk about it in this class. Awesome. Thank you so much. All right. Take care, Zach. Hey. Yes. Yep. The quiz is good. Yeah, we should be good. Yep.
I just had a quick question about AMP regulation.
Yeah. What's up?
Like a while back.
Okay.
Does it bind in that same